Hand sanitizers could damage critical supporting cells in the brain. Chemicals found in common household disinfectants, glues, and furniture textiles could damage supporting cells in the brain during critical stages of their development. A new study based on human cell cultures in mice has found, starting with 1,823 compounds of unknown toxicity found in the environment. Erin Cohn, a molecular biologist at Case Western Reserve University, Ohio, and colleagues identified two classes of chemicals that either killed or halted the maturation of cells called oligodendrocytes under laboratory conditions. Oligodendrocytes are a type of neurological support cell. They wrap around neurons to form an insulating covering that keep brain signals scouting along at speed. One of the two chemical classes identified were quaternary compounds. These are used in disinfectant sprays, wipes, and hand sanitizers, and personal care products such as toothpaste and mouthwash to kill bacteria and viruses, and can be ingested or inhaled if used incorrectly or in poorly ventilated spaces. Another class were organophosphates, serving as flame retardants. They are commonly found in textiles, glues, and household items such as furniture and electronics, and can off gas into the air of rooms we commonly spend time in. Being fat-soluble, organophosphates can be absorbed through the skin and potentially make their way into the brain. In mice experiments, pups gave an oral dose of one of three quaternary compounds, had detectable levels of those chemicals in their brain tissues days later, suggesting that the compounds can cross the blood-brain barrier, the protective fortress between the bloodstream and brain cells. The animals also had depleted number of oligodendrocytes in their brains after 10 daily doses of one particular quaternary compound, cetylpyridinium chloride, administered in a key period of the brain development starting 5 days after birth. Similar effects were seen in brain organoids, clusters of human stem cells grown in a dish and coaxed into behaving like developed brain tissue. We found that oligodendrocytes, but not other brain cells, are surprisingly vulnerable to quaternary ammonium compounds and organophosphate flame retardants, says Cohn. Experts, however, are not immediately alarmed by the results of this lab-based study, which treated cells and mice with concentrations higher than what humans would typically be exposed to, and in ways that do not reflect how humans would come into contact with these chemicals. It's not a question of if something is toxic or not, but if toxic is toxic under the conditions in which we are likely to be exposed, says environmental chemist Oliver Jones of RMIT University, Melbourne, Australia. In this case, the authors have exposed cells in a Petri dish to a relatively high amount of these compounds, which is not the same dose, route, or duration of exposure that humans might encounter normally. However, we know that some people, such as school and hospital cleaners, child care providers, and people in correctional facilities, are more exposed to quaternary compounds than others because industrial strength disinfectants are commonly used in these settings. And what's more, until only recently, research into toxicity of quaternary compounds came from animal and cell studies, but now studies are probing the health effects in humans and finding that people had double the levels of quaternary compounds in their blood during the pandemic than before, likely because of the widespread use of disinfectants. This has led some researchers to flag these chemicals as a class of emerging concern, noting the challenges of fully understanding the human health risks of these compounds due to their vast structural diversity. It's a similar situation with organophosphate flame retardants, which have been widely detected in the environment and in human blood, urine, placental tissue, and even breast milk. Watch out for them to come to the attention of the Stockholm Convention when it's done struggling with organofluorines, PFAS, says environmental chemist Ian Ray, who is an advisor to the United Nations Environment Program on chemicals in the environment and who was not involved in the current research. In their study, Cohen and colleagues analyzed levels of one flame retardant by analyzing levels of one flame retardant metabolite, BDCIPP, in children's urine samples collected in the US CDC National Health and Nutrition Examination Survey between 2013 and 18. Almost all of the 1,763 children aged 3 to 11 had BDCIPP present in their pee, and those with the highest levels were 2 and 6 times more likely than those with low exposure to experience adverse neurodevelopmental outcomes such as motor dysfunction or requirements for educational assistance. But observational data can only point to associations, not direct causes, and large gaps remain in our understanding of how these chemicals affect humans, since, like this study, most of the data comes from animals and cells, and that's reason enough to keep investigating the health effects of these compounds, especially in children. Cohen and colleagues argue the developing central nervous system is particularly sensitive to environmental insults, and chemical exposure can be especially harmful to children if they occur during critical periods of development. This study was published in Nature Neuroscience, and it's on Science Alert by Claire Watson. Please leave your comments, and thank you for your support. Kindly support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.